Hello, I'm Junisa Consuegra. I am the Science Research Assistant of the DNA Barcoding project in Southern Mindanao. The overall objective of our project is to document the reef fish diversity in our study areas and generate their genetic barcodes. This will help us come up with a checklist of reef fishes and a database that contains all local biological information that will be made available for all. We visit different landing sites and wet markets in Davao Gulf and along the island chain of Basilan, Sulu, and Tawi-Tawi. We obtain the reef fishes, perform initial ID based on their morphology, extract the tissue, and preserve the whole fish as voucher specimens. We also record their body measurements and take their photographs as additional biological information. In our fish barcoding laboratory, we extract the DNA from the fish muscle tissue using commercial DNA extraction kits. We then use the DNA as template to amplify the fish barcoding region. This is a fragment of the cytochrome oxidase 1 gene, or COL1, of the mitochondrial DNA. We amplify the COL1 marker using polymerase chain reaction, or PCR. This method makes millions of copies of our target marker. This reaction is made possible by a thermal cycler and, of course, PCR kits. After PCR, we check amplification success using agarose gel electrophoresis. We allow the PCR products to migrate through an agarose gel using electrical charges. Electrophoresis resolves DNA fragments on the basis of their molecular weights. Smaller DNA fragments migrate faster than the larger ones. Using a reference molecular ladder, we can determine the fragment size of our PCR products. After electrophoresis, we view the gel using a gel documentation system. The presence of a bright band with an approximate molecular weight of 650 base pairs tells us that COL1 amplification is successful. We then send the PCR products to a biotech facility, such as the Philippine Genome Center for gene sequencing. Once we receive the sequence data, we prepare them for querying on online sequence databases. The resulting hits from the search serve as the genetic ID of our samples. We then compare this with the morphological ID. Aside from market surveys and laboratory works, we also conduct training workshops for our partner higher education institutions in Northern Luzon and Southern Mindanao. We have also provided internship programs on fish barcoding to senior high school students of the Philippine Science High School in Caraga and Southern Mindanao. We are currently preparing a laboratory manual which details our work. These are the same techniques that we introduce to our partner institutions so that researchers are fully guided and involved.